Today we're looking at one of the absolute most fascinating paradoxes in all of biology. Just imagine, if you will, a complete, fully functional, incredibly complex animal system. It hunts, it breathes, it reproduces, it adapts to highly hostile environments. Now imagine all of that immense biological complexity crammed into just one single microscopic cell. Literally. That is the reality of the protozoa. It completely defies our macroscopic understanding of how life is actually supposed to work. Okay, let's dive into this and begin our biological hardware teardown of these incredible organisms.
Here is our syllabus for today's explainer. First, meet the protozoa. 2. Core biological specs. 3. Movement and fuel. 4. Reproduction and survival. And finally, 5. The cellular blueprint.
All right, let's kick things off with Section 1, Meet the Protozoa.
When we actually look at the kingdom Protista, we're seeing an incredible level of evolutionary efficiency. The word protozoa comes straight from two Greek roots, proto meaning primitive, and zoa meaning animal, primitive animal. And as unicellular microscopic eukaryotes, they are absolutely the undisputed champions of doing more with less. They represent this vital evolutionary bridge where complex, animal-like behaviors, things like actively hunting and reacting to stimuli, happen entirely within the confines of a single cell. Moving right along to Section 2, Core Biological Specs.
If I had to pick just one word to describe the protozoan habitat, it'd definitely be everywhere. Their highly adaptable nature allows them to thrive across incredibly diverse environments. You're going to find them in aquatic environments, both freshwater and marine. You'll find them in the dirt right beneath your feet. And, well, highly relevant to medicine and ecology, you're going to find them living right inside other host organisms, causing massive global impacts. Think malaria or dysentery.
When it comes to their physical dimensions, protozoa operate on a strictly microscopic scale. The standard spec ranges from about 1 to 150 micrometers. To put that into perspective, a single strand of your hair is about 70 micrometers thick. But, biology always has its rule breakers. Outliers like the ciliate Balantidium coli really push these boundaries and get much larger. It just goes to show that even within very strict single-cell limits, there is room for immense physical variation.
Now, what's really interesting about this slide is the architectural choice protozoa make regarding their outer boundary. Unlike plants or many bacteria that rely on a really thick, rigid cell wall for structure, protozoa completely lack a cell wall. Instead, they use a cell membrane, or a highly specialized, flexible outer covering called a pellicle. And this is a brilliant evolutionary trade-off. By dropping that rigid wall, they gain incredible flexibility. They can change shape, squeeze through tight spaces, and move dynamically, all while that pellicle still provides a definite shape and essential armor.
But honestly, the most staggering spec is this. It is a single eukaryotic cell that performs all metabolic activities. Think about it. In humans, we have this massive division of labor, right? We have stomach tissue for digestion, muscle tissue for movement, nerve tissue for signaling. In protozoa, there is absolutely no division of labor across tissues, just one cell doing absolutely everything. And because they're eukaryotes, this single cell operates with a true nucleus, a nuclear membrane, and a nucleolus, keeping its vital genetic code highly organized.
Let's shift gears to Section 3, Movement and Fuel.
To get around, protozoa have developed distinct, highly specialized engines for movement, their locomotory organelles. First up, we have pseudopodia, literally meaning false feet, which give us that classic amoeboid movement you see in things like ant amoeba. Next, we've got flagella. These provide a fast, whip-like propulsion, heavily utilized by Giardia and Trypanosoma. Then there are cilia. These are numerous, coordinated short structures that beat in a steady rhythm, like in Balantidium coli. And finally, in some parasitic forms, well, movement isn't really necessary because the host does the moving for them, so locomotory organelles are just entirely absent. It's also worth pointing out that organisms like Euglena and Trypanosoma rely heavily on that pellicle we talked about to maintain a distinct shape while these engines propel them forward.
Once they move, they gotta eat, right? And their fuel intake methods are incredibly varied. They're largely heterotrophic, meaning they consume other organic carbon. This actually breaks down further into holozoic, where they ingest solid food particles, or saprozoic, where they absorb dissolved nutrients. A lot of them are parasitic, just stealing fuel directly from a host. Autotrophic nutrition, meaning they actually make their own food via photosynthesis like a plant, is actually pretty rare in this group, though organisms like Euglena can cleverly switch it on when they need to. Now, processing all that fuel requires respiration and excretion. For general day-to-day gas exchange, taking in oxygen and expelling CO2, they just rely on simple diffusion right through their body surface. But managing water pressure, that requires specialized hardware. Because water constantly wants to rush into the cells of freshwater protozoa, they employ contractile vacuoles. These act exactly like rhythmic bilge pumps on a ship, actively expelling excess water to maintain osmoregulation. Basically, it stops the cell from bursting.
Which brings us to Section 4, Reproduction and Survival.
When looking at how they continue their species, we see some very distinct strategies, and this brilliantly illustrates the division between everyday replication and specialized processes. For everyday replication, pretty much all protozoans rely on asexual reproduction. That includes binary fission, where the cell just simply splits into two, or multiple fission, known as schizogony, where one cell divides into many simultaneously. But when the situation requires genetic diversity, specialized sexual processes kick in. This includes syngamy, where two gametes fully fuse, conjugation, where they temporarily connect to literally swap genetic material, or sporogony, which is a complex phase of spore formation heavily utilized by sporozoans.
But what happens when the environment turns lethal? The water completely dries up or the temperature spikes? They enter lockdown mode. Quote, formation of resistant cysts for survival and transmission. This process, called encystment, is the ultimate system backup. The protozoan essentially builds a tough protective wall around itself, shuts down its metabolism, and just waits. In this cyst form, it can survive extreme hostility and be transmitted to entirely new environments or hosts, only reactivating when conditions are safe again. It's an incredible survival mechanism.
Finally, Section 5, the cellular blueprint.
So zooming physically past the pellicle, we find that the cytoplasm isn't just some random disorganized soup, it's highly, spatially organized into two distinct zones. You have the ectoplasm, which is the outer, clear, slightly more rigid layer right beneath the membrane, and the endoplasm, which is the inner, more fluid, granular layer. This spatial organization is critical. It keeps internal cellular processes strictly structured, ensuring that the locomotion mechanics happening on the outside don't interfere with the complex digestion and genetic processing happening on the inside.
So, the crucial point is how they actually manage their genetic code, particularly when we look at ciliates. They utilize this phenomenal adaptation called nuclear dimorphism. Guys, they literally have a dual processor system. They maintain a large macronucleus that handles all the everyday vegetative functions, metabolism, protein synthesis, the daily grind. Then they keep a separate, smaller micronucleus that is kept totally pristine, entirely reserved just for reproductive functions and storing the master genetic backup.
Deep down within the endoplasm, we find the specialized vacuoles. Now, we already touched on the contractile vacuoles, those rhythmic pumps keeping the cell pressure regulated in freshwater, but working right alongside them are the food vacuoles. Because there is no stomach, when a protozoan ingests a food particle, it just wraps it in a membrane to create a food vacuole on the fly. It's a temporary, mobile digestion center where enzymes break down the fuel right there inside the cytoplasm.
And finally, floating within this complex cellular blueprint are all the standard eukaryotic organelles you'd naturally expect to see in much higher organisms. Ribosomes for protein synthesis, the Golgi apparatus for packaging those proteins, and the endoplasmic reticulum for transport. They also have mitochondria to generate power. But what is truly amazing here is that in anaerobic protozoa, the ones living in environments completely devoid of oxygen, these mitochondria are dynamically modified into specialized structures, like hydrogenosomes or mitosomes, perfectly tailored to their specific low-oxygen environmental specs.
We've seen how they move, how they eat, how they survive, and the staggeringly complex machinery operating inside them. It really makes you completely rethink the definition of simple life. I mean, if a single microscopic cell can engineer flexible protective armor, run on a dual-processing nuclear system, and build varied, highly specialized locomotory engines, what other complex limits can a single cell push? Keep that question in mind as you continue to explore the absolute perfection of advanced microbiology. Thanks so much for joining me on this teardown, and I'll see you in the next explainer.